All right, so hello everyone and welcome to our webinar, The Discovery of HIV, 40 Years of History, Science, and Controversy, hosted by Wiley and the American Society for Microbiology. I'm Megan Angelini, Managing Developmental Editor for ASM Books, and we're really pleased that you're joining us today. As a note, you're in listen-only mode to cut out any background noise. However, we do invite you to type your questions into the Q&A box at any time so that the presenter can address them during the Q&A portion of the webinar. This session is being recorded, and in a few days' time, you'll receive a follow-up email with a link to the recording. Now, please allow me to introduce today's speaker and author of the book, Germ Theory, Medical Pioneers in Infectious Diseases, second edition, Dr. Robert Gaynes. Dr. Gaynes is a former chief of the surveillance activity of the CDC's Hospital Infection Program, a former attending physician and chair of the Infection Control Committee, Antimicrobial Stewardship Committee, and COVID-19 Vaccine Planning Committee at the Atlanta VA Hospital, and a professor emeritus of medicine at Emory University School of Medicine, where he continues to teach the history of medicine. We're so happy to have him here for this webinar. And so without any further ado, I'll hand it over to Dr. Gaynes. Thank you.
Well, thank you very much, Megan. And it is always a pleasure to be here and to work with the people from ASM. As many of you may know, 2023 represents the 40th anniversary of the discovery of the human immunodeficiency virus, or HIV. One of the most important pathogens of the 20th century, and arguably one of the most important pathogens ever discovered. Today, we think nothing of sending a blood test off for a patient who might have HIV infection. And if they have HIV infection, we have a variety of different drugs to treat them. And when you consider that in the 1980s, the syndrome of acquired immune deficiency syndrome, or AIDS, which was a clinical diagnosis at the time, carried a nearly 100% mortality in about 4 years. And today, with these drugs that we have, if a patient takes their drugs and has a suppressed viral load, they have a lifespan that is nearly the same as someone who's never had HIV. And these two amazing changes in the natural history of this syndrome are owed to the discovery of the virus HIV. However, its discovery was laden with a tremendous controversy, unlike perhaps any other in the history of science. So given that this is now the 40th anniversary of that discovery, I thought it might be time to kind of review how that all came about and what was the genesis and eventual resolution of this tremendous dispute. Well, to begin with, we have to go back to the beginning of what we consider the acquired immune deficiency syndrome. And in June of 1981, in the CDC's Morbidity and Mortality Weekly Report, or MMWR, was the first report where the scientific community at large became aware of this unusual cluster of opportunistic infections, pneumocystis pneumonia, among 5 homosexual men in Los Angeles, California. Now, many people thought this was just a fluke and that there was no particular explanation. But within months, another report came of a particularly unusual sarcoma, a type of cancer that occurred generally in very old men or people who had transplants. And yet this was also occurring primarily in homosexual men. And it was called Kaposi sarcoma, and it was a rather aggressive form that had never been seen before. More cases of pneumocystis and Kaposi sarcoma began piling up in 1981. And in 1982, epidemiologic evidence strongly suggested that this clinical syndrome of AIDS was, in fact, an infectious disease, that it was transmitted sexually, And whatever the agent was could also be transferred by exposure to contaminated blood or blood products. In fact, the blood supply could no longer be fully safeguarded until the agent was found and a test to measure for the agent in the blood supply was performed. Unfortunately, an entire generation of hemophiliacs, some 15,000 in the United States, became infected because of contaminated blood products that were needed to supply them with the various clotting factors. This generation of hemophiliacs all got AIDS, but was, as you'll see, one of the first clues of what the agent was when people looked at what was going on. During 1982, evidence for transmission from mother to child was also reported, and sharing of contaminated needles among IV drug users helped further spread AIDS. But with all of this going on, no one knew what the agent was. The first clue came not from virologists, but from immunologists. It was noted that patients who had this clinical syndrome of AIDS had a dramatic decrease in a certain type of white cell. It was a T lymphocyte, and not just any T lymphocyte, but a T lymphocyte with a marker called the CD4 marker, or the CD4 cell. And I've likened this to the immunologic response of humans being a symphony. And the CD4 cell is the conductor of that symphony. While many aspects of immunologic response were still present in AIDS patients, it was a disorganized approach to immunology, much like a symphony trying to play a concerto without a conductor. So the CD4 cell was a major player in what was going on in these patients, and whatever the agent was going to have to account for why the CD4 cells were disappearing in AIDS patients. The other aspect, which I mentioned a moment ago, was the fact that hemophiliacs were all getting AIDS. And this was a particularly important part of the etiologic search for this microorganism, because the only microorganism small enough to evade the filters that were used to screen clotting factors was a virus. So it suggested very strongly that not only was there an etiologic agent that was a microorganism, but it was likely some sort of virus. The picture here is of Ryan White, who was in fact the poster child for hemophiliacs with AIDS. He, unfortunately, like all the others of his generation, got AIDS and had an opportunistic infection. He had pneumocystis and was hospitalized. When he recovered from the pneumocystis, he was prohibited from attending school. And unfortunately, it's a horrible reminder of the incredible and ugly intolerance that patients with AIDS and even patients who were in various risk groups for patients getting AIDS experienced during this rather difficult period in the 1980s. He's pictured here with Elton John. Well, in early 1983, an article appeared in the Journal of the National Cancer Institute that suggested that there were 3 possible candidates of viruses that were the most likely culprits. The first suggested that it was a variant of the hepatitis B virus, and that was largely because many of the risk groups for AIDS were very similar to the risk groups for patients with hepatitis B. However, it was noted in the article that none of the patients with AIDS seemed to have major disruptions of their liver, unlike patients who had hepatitis B. That was largely discarded. The second group that was considered was a herpes virus, herpes simplex, herpes zoster, and especially cytomegalovirus, or CMV. In fact, if you look back in late 1982 and early 1983, you can actually find articles that tout CMV as, in fact, the cause of AIDS. However, this was also discounted because of the irregular findings and the fact that CMV was not more common in AIDS patients than it was in other individuals, since many adults have been exposed. And the final group of viruses that was considered in this article were the retroviruses. Now, it's important to kind of go back and understand the history of the retrovirus. That history actually begins with the discovery of an enzyme, reverse transcriptase. This was discovered in 1970 and has been described as one of the most significant scientific discoveries in the second half of the 20th century, because it overturned a central dogma of genetics, that information in genes flowed from DNA to RNA to proteins, and never the reverse. The reverse transcriptase, however, began to upset that dogma. The history of that goes back to a man named Howard Temin. In the 1950s, Howard Temin was working with the Rous sarcoma virus, an RNA virus that was capable of inducing a certain kind of cancer, sarcoma, in chickens. His experiments suggested that the viral RNA was being converted in the host cell into DNA and inserting in the host cell genome in violation of the central dogma. That is, the RNA was going into DNA and going into the host genome. Temin called this conversion of viral RNA into DNA a pro-virus, and no one believed him. For over 15 years, he touted this, but was not only not believed, but often ridiculed for his ideas. But finally, in 1970, Temin found the enzyme reverse transcriptase that was responsible for making this so-called provirus and showed that it was able to convert RNA into DNA. Working with a different RNA virus, a man named David Baltimore discovered the same enzyme at virtually the same time, bolstering Temin's work. And they validated the idea that these viruses, that were now called retroviruses, could cause cancer in animals by incorporating into and then altering the DNA of the host cells. Along with Renato Del Becco, Temin and Baltimore shared the Nobel Prize in Physiology and Medicine in 1975 for this important discovery. The discovery of reverse transcriptase and retroviruses in animals set off in 1970 a 10-year search for retroviruses in human beings. Unfortunately, that search was largely futile. The only human cancer that is known to be directly caused by a retrovirus, the human T-cell lymphotropic virus, or HTLV1, was discovered in Robert Gallo's lab at the National Institutes of Health. More on that in a moment. Now, circa the 1970s, how did people look for a human retrovirus? There were basically 4 steps involved. And this is obviously oversimplifying it, but you had to choose the correct human or animal cell line that could be cultured in the lab and maintained in the lab. Not an easy business. You had to find a source for the putative virus that could, in fact, infect this particular cell in cell culture. Once that was done, the third step was a detection method for the virus that was available after the cell line was infected, which was essentially detection of this reverse transcriptase as possible evidence that there was a virus involved. And finally, you needed a means to isolate and characterize the virus from these cells. And that's how the search went on. Well, there were 2 major advances that occurred from Dr. Gallo's lab during this period. First was a technique to detect reverse transcriptase, which was not an easy feat. There were many enzymes in the cell of humans and animals that make this a very difficult process. And he described a technique that actually made it much easier and more accurate. The second advance from his laboratory was the idea of maintaining T lymphocyte cultures in the laboratory by the discovery of the T-cell growth factor. Later, this has been named interleukin-2 or IL-2, and the presence of IL-2 in T lymphocyte cell cultures made maintaining these cell cultures much, much easier and efficient. Well, using these techniques, Gallo and his colleagues were able to isolate the first human retrovirus, which was first named the human T-cell leukemia virus. Now it's called the human T-cell lymphotrophic virus, or HTLV-1. HTLV-1 was isolated and associated with a human T-cell leukemia. And pictured on the screen, you can see the morphology of HTLV-1. A relatively uniform morphology, as you can see on the screen, and I'd like you to keep that in mind as we move ahead in our discussion. In 1982, Gallo and his co-workers discovered a second human retrovirus, which was named HTLV-2. It was in a patient with a disease called hairy cell leukemia. However, other patients with hairy cell leukemia did not seem to have HTLV-2, and in fact, no other malignancy or other disease has been clearly linked to HTLV-2. Only HTLV-1 has been clearly associated with a human T-cell leukemia. Well, there were very few laboratories that were capable of doing this kind of work. Obviously, the one at the NIH, and another one had been set up at the Pasteur Institute in Paris. And here we enter that scene. One of the key individuals involved is a woman named Françoise Barré-Sinoussi. And in June of 2021, I had the opportunity to interview Professor Françoise Barré-Sinoussi by telephone. Many of the comments I'm about to make, and in fact, a few audio clips from Professor Barré-Sinoussi came from that interview. I also refer you to further detail of her career and much of what else I'm about to say, because I can only present so much at our webinar, in chapter 15 of the second edition of my book, Germ Theory. In addition, I did a podcast with ASM, and the podcast is available with more audio clips than I'm able to put into our talk today. Now, Professor Barré-Sinoussi was born in Paris in July of 1947. She was quite good at the natural sciences and attended the University of Paris, but after 2 years of study, she wanted to get more practical experience. She looked around with the help of some friends and was refused jobs at a variety of laboratories until a friend asked her boss, Jean-Claude Schiermann, if he would consider hiring her. And he did, initially on a part-time basis, and eventually she was hired on a full-time basis. Jean-Claude Schiermann eventually would become the head of the retrovirology unit at the Pasteur Institute. He helped her get her PhD in 1975, and after she received her PhD, she did a postdoctoral fellowship at the National Institute of Health in Robert Bassen's lab, but that period included an entire month working in Dr. Robert Gallo's lab, and she made acquaintances with him and others at that time. In 1976, she returned to France, and upon her return, she continued to work on murine retroviruses in the same laboratory as she had left. But Jean-Claude Schiermann had moved up in the organization, and the unit was now run by a man named Luc Montagnier. He was studying the link between an oncogene and cancer and was not specifically interested in retroviruses. But Françoise Barré-Sinoussi had her own lab where she isolated retroviruses in the work. And she did much of this work in her research without really garnishing much attention. But that dearth of attention was about to change. In December of 1982, clinicians in Paris met with Jean-Claude Schiermann, Luc Montagnier, and Françoise Barré-Sinoussi, and this is where she first learned about AIDS, its epidemiology, the symptoms, and the at that time, overwhelming mortality. The clinicians believed that there might be a retrovirus involved in this, And the doctors gave Montanier's lab a sample from a lymph node biopsy that they had taken with a patient with something called generalized lymphadenopathy. Now, this is a syndrome in which the patient has lymph nodes that are somewhat enlarged, usually not tender, and it exists for months to a few years before the patient develops AIDS. It was a syndrome that was generally recognized to be a precursor of AIDS. Now, from this lymph node biopsy, Montagnier cultured the cells from the lymph nodes using techniques pioneered, as I mentioned, in Dr. Gallo's lab. After the lymph node cultures were set up, a day or so later, the cell cultures were brought to Barré-Sinoussy's lab to look for the presence of reverse transcriptase in the procedure I described a few moments ago. Well, Barré-Sinoussi sampled the cell cultures using several methods. She became quite adept at this to detect the presence of reverse transcriptase as an indicator that there might be a retrovirus present. And after 2 days, she began to find evidence of this enzyme, but it was at low levels, and she decided not to mention it to anyone. So for 2 days, she was the only person that knew that there was likely a reverse transcriptase in this sample. 2 days after that, the level of the reverse transcriptase was high enough that she was certain of its presence, and she told her collaborators that there was evidence a retrovirus was present in these cell cultures. Unfortunately, calamity occurred 2 days later. The cells that were producing this enzyme began to die. The researchers needed more viable lymphocytes in cultures, or whatever this virus was going to be lost. Fortunately, the Pasteur Institute had a blood bank, and Barré-Sinoussi and other researchers went over to the blood bank and got normal lymphocytes, and they added these fresh lymphocytes into the cell cultures with those dying cells containing the putative virus. And much to her amazement, after about 2 weeks, Barré-Sinoussi could again detect significant levels of reverse transcriptase, and those levels remained stable for several weeks. Now, this cell death in cell cultures offered a vital clue. One of the differences between these infected T lymphocytes that Pasteur Institute was maintaining was that, unlike in HTLV1, the T lymphocytes with HTLV1 proliferated. Rather, the T lymphocytes in Barré-Sinoussi's culture died. And it turned out this finding was quite consistent with clinical observations of diminishing CD4 cells in AIDS patients, which was the primary target of whatever this virus, putative virus, was. The researchers performed experiments successfully distinguishing this new virus from HTLV1, and they named the new virus lymphadenopathy-associated virus, or LAV. You can see the collection of individuals involved in this. Once they had determined this, all of the work that they had been doing on murine retroviruses was stopped, and they turned all of their attention to this new virus. Barré-Sinoussi sent samples to colleagues at Pasteur who took pictures of the infected lymphocytes using an electron microscope. And what you're seeing on the screen right now is the very first image of the lymphadenopathy-associated virus, or what, as we'll see, is the HIV virus. And if you look carefully, you can see there are some morphologic differences between what I showed for HTLV1 and this particular virus. You can see that by the arrow on the screen. Well, the group actually initially hurriedly prepared a paper and sent it to the journal Nature, and it was rejected. They took some of the comments and sent the paper to the journal Science. In May 20th of 1983, their article was published. In that article, the authors wrote, "The role of this virus in the etiology of AIDS remains to be determined." Now, you'll remember that there was very little data from other patients, in this particular article. And this was from a patient who did not actually have AIDS, but had what was called pre-AIDS. And I remember asking in our interview, Professor Barré-Sinoussi, how sure she was that they had actually found the cause of AIDS. Did they know it or not? And here is what she said.
Yes, we were sure we had it, but we could not really write that, because if you want to make sure that the virus was a cause of a disease, you had to accumulate evidences that we did not have ourselves in hands at the time. So we could not write that was the cause of AIDS, but we were sure that was the cause of AIDS.
In fact, I presented this talk at Emory University, and sitting in the audience was the former director of the Center for Disease Control when all of this went down. And he was giving a talk at the time as well. And after the seminar session was over, I went up to him and asked him if I had said anything that he was unaware of when he was working. And he told me that, and he used this word, he was flabbergasted as to how sure the French were right from the beginning that they had found the cause of AIDS.
Yes.
Well, not everyone agreed, unfortunately, that the French had found the cause of AIDS. The French paper that was published in May of 1983 received very little attention over the next 6 months, only 27 citations in scientific journals from this paper. Now, you'll know now that that was, in fact, the first paper that described what we now know of as HTLV, or as HIV, rather, but it really didn't seem to catch very much attention. And the reason for that is in the same issue, the May 20 issue of 1983 of Science, there were other reports that suggested an association with the syndrome of AIDS and HTLV-1 infection. And these papers were from Robert Gallo and Max Essex, who were very big experts in retroviral infections. And many people believed that, in fact, it was HTLV-1, the virus that Bob Gallo's lab had isolated, that was the cause of AIDS. I asked Professor Barré-Sinoussi about this.
We were aware about what's going on in Bob Gallo's lab because we were in contact with Bob Gallo. Since the identification of our virus, one of the first things that Jean-Claude Sherman did was to call Bob Gallo after a year before, in 1983, because we wanted to have a reagent in order to compare the virus we just isolate to HTLV-1. So the reason why Jean-Claude Sherman called Bob Gallo, and we had continuous exchange between Bob Gallo and our lab since 1983. So we knew what was going on in Bob's lab, and we had even, Bob was completely, we had a long discussion. I went also to Bob Gallo's lab to make a presentation, by the way, in 1983. And at the end of my talk, we had this discussion with Bob. Bob was telling me, Françoise, I'm sure it's an HTLV. And I said, no, Bob, it's not an HTLV. We were.
Well, the controversy started to rise. Montagnier and François Barré-Sinoussi were busy characterizing the virus they called LAV and its association with AIDS. They were, in fact, collaborating with people in Gallo's lab, with the CDC and others. But towards the end of 1983, Gallo's lab went silent, and they were secretly beginning to change their focus from saying that this was HTLV-1 that was the cause of AIDS to a broader search for a retrovirus distinct from HTLV-1 as the cause of AIDS. And in fact, no one was really aware in the last few months of 1983 into early 1984 of what Gallo's lab was doing until April 23rd of 1984, when Margaret Heckler, who was then the secretary of the Department of Health and Human Services, held a press conference.
Good afternoon, ladies and gentlemen. First, the probable cause of AIDS has been found, variant of a known human cancer virus. Second, not only has the agent been identified, but a new process has been developed to mass produce this virus. Thirdly, with discovery of both the virus and this new process, we now have a blood test for AIDS. With the blood test, we can identify AIDS victims with essentially 100% certainty.
Thank you, Secretary.
Now, Heckler added later in the press conference that all the credit must go to our eminent Dr. Gallo, and she predicted that an AIDS vaccine would be available within a year or two. Remember, this was 1984. 10 days after this press conference, an article appeared in the May 4th issue of Science in 1984. Gallo and his colleagues reported the discovery of a new virus that he called HTLV-3. Antibodies to this virus were detected in almost all the patients with AIDS. The virus could be isolated from a fairly large number of patients with AIDS and those with this unexplained lymphadenopathy or pre-AIDS. And Gallo's lab was able to grow large amounts of this virus in a specialized cell line of T lymphocytes. In this article, Gallo and his colleagues suggested that HTLV-3 and the French LAV were different viruses, but noted that, quote, it is possible that this is due to the insufficient characterization of LAV. And the debate began. This debate had huge implications. This was a very important virus causing a very important clinical syndrome that was now affecting tens, if not hundreds of thousands of people. And first, who should get the credit for finding the cause of AIDS? And second, each laboratory had developed a blood test that was designed to detect antibodies to this newly discovered virus. And this test would be worth millions of dollars, both for diagnosis and for testing blood to keep the blood supply safe. So the implications of who actually found the virus that caused AIDS were enormous. Now, the controversy then exploded because one day after that press conference, Gallo applied for a patent on his antibody test. Now, the Pasteur Institute had applied for a U.S. patent on their antibody test a full 4 months before Gallo. And in May of 1985, the U.S. Patent and Trademark Office granted Gallo the patent on his blood test, which was then used to detect antibodies to this virus. A few months later, in December of 1985, the Pasteur Institute filed claims against Gallo's claim in U.S. Claims Court, and the controversy exploded. In fact, the controversy was a clash, as one journalist called, of two titanic egos. Both Gallo and Montagnier, according to many reports, had such egos that they simply could not accept the fact that another lab might have been correct. In fact, the two were so in clash over this that they refused to be in the same room at conferences. The dispute became very bitter and began to be linked, actually, to scientific prestige of each nation, and it strained relations between both labs, and particularly, as I mentioned, between Dr. Gallo and Montagnier. Now, I asked Professor Françoise Barré-Sinoussi about this, and she told me that she tried very hard to stay out of this clash. She had worked in Gallo's lab, she knew some of the people that were working there, and she continued to try to collaborate with members of this lab until the dispute became so bitter that it prevented any continuation of any partnership. Eventually, this resolution of this festering controversy went all the way up to the leaders of the two countries, the President of the United States, Ronald Reagan, and the Prime Minister of France stepped in to end this conflict in 1987. The two countries agreed in an extraordinary settlement in science, I think perhaps even a unique settlement in science, to a 50-50 split of the credit and the patent royalties from the work with the virus and the antibody blood test. Unfortunately, it didn't end there. In early 1985, a DNA analysis of the French and American viruses showed that they were virtually identical. And if you know anything about what we now know of as HIV, you'll know that this virus continually mutates. And the fact that the DNA analysis showed that the French and American viruses were virtually identical led to a variety of accusations being hurled at Dr. Gallo that he might have, and I'll use the word that was used at the time, misappropriated the French virus since they had been sharing samples. Well, this led to a very convoluted investigation, first by the NIH and then by the U.S. Department of Justice. Several years into this, Gallo gave a scientific explanation of why his virus sample was so much like the one from Luc Montagnier and Barré-Sinoussi's lab. He said that it was contamination, not theft, as the explanation. And finally, after 4 years of investigation, the U.S. government eventually dropped all accusations of misconduct against Dr. Gallo in 1993. Now, Jay Levy and his colleagues from the University of California, San Francisco had independently isolated what turned out to be the same virus, which they named the AIDS-associated retrovirus, or ARV, in 1985. And in May of 1986, the International Committee on Taxonomy of Viruses proposed calling the virus that caused AIDS the human immunodeficiency virus, or HIV, which is how we know it today. Well, when it became clear that the U.S. patented HIV blood test was based on a laboratory contamination from a French virus, the deal between the French and the U.S. over patent royalties was renegotiated. And finally, in 2003, Drs. Gallo and Montagnier seemed to have settled their disagreement and co-wrote a perspective article in the New England Journal of Medicine entitled "The Discovery of HIV as the Cause of AIDS." In 2008, the Nobel Prize Committee weighed in on who they felt should get the credit for the discovery of this virus. And along with Harold Zuhausen, who discovered the human papillomaviruses causing cervical cancer, half of the Nobel Prize went to François Barré-Sinoussi and Luc Montagnier. In his 2008 Nobel Prize address, Montagnier said that both Jean-Claude Schirman and Robert Gallo should have been allowed to share in the Nobel Prize. In 2022, Luc Montagnier died. Now, there are many lessons that can be drawn from this early period, but I will quote from that perspective article in the New England Journal, quote, "Many lessons can be drawn from this early intense period, and most suggest that science requires greater modesty." I thank you for your attention.
Thank you so much, Bob. We're now going to move on to the question and answer portion of the webinar. So feel free to type questions into the Q&A box on your screen. We've had some great questions come through, and we'll try to get to as many as possible. So the first question I have here is, Bob, can you speak a bit on whether the controversy's focus affected patient testing and health outcomes?
Actually, I don't think it did. The controversy really allowed more the credit and the royalties more than the actual testing. Testing went forward fairly quickly, and there was not a major controversy over the testing.
Interesting, thanks. Another question I have here is, can you speak a bit on and maybe compare sort of the early days of the HIV pandemic with the early COVID-19 pandemic?
Well, they were very different. And most of that is because of the means of transmission of those two viruses. Essentially, and I remember giving talks about this to people, essentially in the 1980s, with certain behavioral approaches, you could rather easily stay out of the way of HIV. The disease was not transmitted casually, although a lot of people thought it might be, but it really wasn't. And there was strong evidence against casual transmission. And of course, in COVID, transmission can occur, and very silently. Patients who are very clearly asymptomatic in a day or two before they become symptomatic can transmit COVID, and it is much more easily transmitted through the respiratory route, which was not true for HIV. And I used to say to people that it was relatively straightforward to stay out of the way of HIV. Obviously, there were certain behaviors that put you at risk, and some of those behaviors were difficult to change in people, but it was a very different transmissibility than what we have seen with COVID.
All right, next question. Where do you think the optimism that there would be a cure or a vaccine in 2 years' time, where do you think that came from?
Well, if you look back at a lot of discovered viruses over the course of the 20th century, most of them, the immune response was such that once antibody started to appear, the person would become immune. That was clearly not true. In fact, one of the bases for the blood test for HIV initially was presence of the antibody, but the antibody is not protective from HIV doing its thing. So that was one of the real difficulties that continues. It's been 40 years, and we still do not have a vaccine for HIV. A lot of people have worked on it. This has been the source of great effort, but the way this virus incorporates into the human genome is one that has really profoundly affected our ability to develop a vaccine. And I think it's the basic molecular biology of this virus, which is this group of viruses. The retroviruses are much different than what we've dealt with most other viruses. And I think that's the major reason, and a good example of how quickly vaccines, in generally pretty effective vaccines, can be developed is our recent experience with COVID. It did not take long to develop a fairly effective vaccine for this, whereas a lot of efforts have gone into developing a vaccine for HIV. And I think it was overly optimistic of Margaret Heckler to have made that pronouncement in 1984. A very different molecular biology of this virus.
Another sort of interesting, similar comparison question for you. If the HIV controversy was happening now for the first time, how do you think, in 2023, our modern world would have shaped how that controversy played out?
Wow, that's a fascinating question, because we have seen, even with a vaccine for COVID that has been profoundly effective by comparison to a number of other vaccines that we have, people have doubted it. And a lot of that is due to social media and the spread of misinformation. And I think using that, if you will, as a model, the chances are much greater that much more misinformation would have been spread if the controversy that we saw in particularly the 1980s had occurred now, some 40 years later. I think the ability for people to spread misinformation is much different than it was then. And I fear that there would have been a lot of other opinions that would have grabbed hold. And unfortunately, I think that is one of the difficulties of our present society. Information can be passed very quickly, but it isn't always correct.
A lot of questions come in with varying wording, asking you to talk a little bit about the Nobel Prize.
So the way Nobel Prizes are awarded is a bit secret. So you don't really know what went on in making this decision. But with the development of hindsight, I think that we can look back and see that, in fact, one, what Dr. Gallo's lab had and what the French had were essentially the same virus, and that the virus was, in fact, discovered in the Pasteur Institute ahead of anything that was reported from the NIH. So in that sense, that's usually where the Nobel Committee weighs in terms of who discovered it first. But in my talk, you'll remember that it was only 5 years from the discovery of reverse transcriptase by Howard Temin and David Baltimore that they received the Nobel Prize. Think of how much time passed between the awarding of the Nobel Prize for this HIV discovery in 2008 and when this controversy really occurred, primarily in the 1980s. It was quite a lengthy period of time. It's an extraordinarily important discovery. Obviously, the virus itself not only has a huge human cost, but it scientifically presented and continues to present major challenges. And so its discovery was really an important element in the I guess the Nobel Committee really wanted to get it right. And I do think that they did in terms of who discovered it first.
All right. Seeing a couple of questions sort of related about this. So you mentioned the lesson that Doctors Gallo and Magnetier wrote, that science requires greater modesty. So are there any other lessons that can be learned in that?
There are plenty of lessons, and I think we could go on for a while about this, but I think one of the major lessons that we should learn from this whole development is the importance of basic scientific research, because that gave investigators the technical and conceptual tools to find the cause of AIDS. And you really have to remember, although it took a while for the Nobel Prize, the discovery of the virus was only about 3 years into the epidemic. And that's really a remarkable period, a short period of time. And I think the tools that were there, things like ways to characterize the enzyme reverse transcriptase, how to grow cell lines, all of that were basic scientific tools from the laboratory. And that was a real payoff during the early days of AIDS in finding this virus. And the second thing I think we should keep in mind as lessons from this is effective collaboration of clinicians, basic scientists, and epidemiologists. That's a really important aspect and was evident in that clinicians thought they might have something and met with basic science researchers in France. And they collaborated on this to find what we now know of as HIV. And I think remembering the importance of that kind of collaboration is really key. And it's something that we need to remember all the way from basic science, all the way through clinical medicine to the epidemiologic side, that good collaboration is really an important aspect of progress.
Thank you. So here's a question about Dr. Barre-Sanusi's Nature rejected paper. So when you interviewed her, did she give any background on what the feedback was from that initial paper? What was the rejection?
It wasn't. Yeah, I think that's a very good question. And it wasn't a major focus of our interview, but she seemed to indicate, and I think others have said this, but that to say that this virus, which at that time was, you recall, called the LAV virus, was the cause of AIDS, had a couple of issues that required a little imagination. The first is that it was isolated in someone who didn't have AIDS. He had what we characterize as pre-AIDS. He had this lymphadenopathy. And was that really AIDS that was the virus involved there? And the second was that they rushed this in, I think in retrospect, perhaps correctly, but they rushed this into publication that they had found this virus, but they had very little data from other patients. Very little data on that first publication from anyone with AIDS. And they worked on that over the course of the next few months. But that initial investigation didn't include anyone who had at that time, it had to be clinically diagnosed, who had the clinical diagnosis of AIDS. And I think that may be, although we will probably never know, why Nature rejected it. But clearly within a month or so, it was a major journal, of course, Science had accepted it. But I don't know that we can say any more than that.
All right, this is a pretty broad question here for you, Bob. So in your estimation, what do you think will be the next agent of viral pandemic? And do you think that we'd be able to cooperate internationally to overcome it? Or was sort of the international collaboration that we saw for COVID-19 from lessons we learned, or kind of a fluke?
Well, it's always hard to know. I think from the story of HIV, some of that was driven by the personalities of the individuals involved. And, we like to think that science is devoid of effects of personality, but from the work, and you can see that in other stories in my book, Germ Theory, personality sometimes has a major role in how things are accepted, or in some cases, not so quickly accepted. In science. So I think there is some aspect of that that always has to be taken into account. But it also has to do with the serious nature of a disease, and how important it is in terms of how much of society and what parts of society are being affected. Unfortunately, for people with what we now call HIV, the initial risk groups that were involved with this virus were on the fringes of our society. And for that reason, they perhaps did not get the kind of attention. The disease itself did not get the kind of attention that it might have should other parts of society have been affected, or if transmissibility was different than it was. I think we should all be concerned about the possibility of another pandemic. Knowing where that's going to come from is quite a challenge for anyone. And I think, the fact that we just recently went through one does not keep us safe for the next 100 years, as we saw with the 1918 influenza pandemic to what we've seen with COVID. I don't know that we can say we'll be safe, particularly as we forage into other areas of the world where we may run across viruses that we so far haven't seen and therefore haven't got immunity to. So I think it's almost an unanswerable question, but there are elements that we can look at for both of these epidemics that we can at least learn some things from.
All right, I'm seeing a lot of questions in the chat in different forms asking about the potentiality for an HIV vaccine. What will it take to get a functional vaccine? What makes it so difficult to develop?
Well, that's the $64,000 question for this disease. Remember, it's been 40 years since we've discovered the etiologic agent of this epidemic. And we're not much closer than we were with the first trials. So I don't know that someone like me can really give a good answer to that question. Part of the reason is the way this virus incorporates its genome, and often very quickly, into the human genome, unlike what we've seen with other, and again, COVID might be an example, where you're not seeing this kind of incorporation into human DNA. And it has made the development of a vaccine for HIV an extraordinarily difficult process. And unfortunately, I'm not the one that can really cite current aspects of that as to where we're going and how soon it will be. This is one that you'd really need to get somebody who's deep into this. I have talked to Mark Mulligan, who at the time was working at Emory. He's now at the New York University, and he's been a major proponent of trying to develop a vaccine, but it has been quite a difficulty. And at the time I talked to him, which was, I think, around, it must have been about 2012, he said that it was going to take 10 to 15 years. And that's been 10 years ago, and we're no closer. So I don't know what it's going to take to do. It's quite a conundrum. And a lot of it is the molecular biology. This is a very different virus than what we're used to in even with COVID, but with influenza, et cetera. There are changes that all these viruses can have, but by and large, it's more the way this incorporates into the human genome that has presented the greatest difficulty in developing some sort of vaccine. And we may be looking at some other approach that is wholly new that will stop people from getting infected. The good news in all of this, however, is that our therapy, if someone is infected, has improved to the point now where somebody who has HIV and is on therapy with a suppressed viral load has the same lifespan as someone who's never had HIV. Unfortunately, at least at this point, if you stop your therapy, the virus gears up again and you're back to where you were when you were infected. But therapy itself has improved dramatically in the last 20 years.
Thanks. All right. I think this next one is going to be our last question since we're getting close to the hour. So this question asks, was there an explanation found for how the contamination occurred in the Gallo lab?
Well, it isn't clear to me. I think you might better ask somebody who was actually in the laboratory as to what it is. There was this is a tricky virus to work with in the lab. So I would say that in some ways I can support the idea that contamination could easily have occurred unknowingly in the laboratory. How it occurred, I don't know. And someone that worked in this area might be able to give a better answer than I.
All right. So we're getting close to time now. So I just want to take a moment to thank Dr. Gaynes for today's excellent presentation and discussion, and to thank the audience for all of your thoughtful questions that you sent in and for taking the time to join us today. As a reminder, a recording of this presentation will be mailed to you within a few days. And with that, we're going to thank you and close out the webinar.
Well, thank you very much for having me.